or somewhat.
Need some lunch.
It's not
I
time
need
for
some
lunch yet.
ATP.
Good use of ATP.
Mhm.
Well, you are eating some, you are taking some snacks. I see some of you have snacks. So that will give you some ADP, especially when you have some sugar in them. So that will give you very quick ATP. What else actually people use for quick energy other than sugar? Sugar is just bad.
Coffee?
Pardon?
Coffee?
Coffee. Coffee just shakes
Energy
you up.
drinks.
Yeah.
Coffee just shakes you up and shakes you up. So people actually use bananas, right?
Yeah,
Bananas.
bananas got a lot of sugar.
Why is that? Why do people use bananas?
It has a whole bunch of sugar in it. I mean, a bunch of carbohydrates.
Carbohydrates, but carbohydrates take some time to be available.
What is it with bananas?
Potassium.
It has potassium.
Potassium is why actually they are considered as quick source of energy because what you just saw, right? In those slides where the potassium is going in and out of the cells through those channels, so it can actually give you very quick energy. Coffee just, I don't know. Coffee
Coffee works.
coffee works too. What if you actually don't have that much sugar in it? Will it still work?
Tastes nasty.
It is actually a stimulant. It's a brain stimulant.
harder.
Yeah. So it will work without it too. The darker it is, more effective it is.
Yes.
Actually, even in those energy drinks too.
Monsters, I used to drink.
Monsters and those
Coffee and two
what is it
Monsters.
called?
5 hour energy shot.
5
Yeah,
energy,
those
yeah.
5 hour.
Those ones, they actually give you a good shot of energy. Has anybody tried those?
Nope.
They make
Either?
They give me jitter.
Do they really work?
Yeah, but you have a crash.
Yeah, you
do have a crash. Yeah, and then it's just like, what did I do?
That's right.
Just lay there.
So you feel guilty after? Yeah, they are just all stimulants. So they will wake you awake and shake you up. Okay, so just before the break we talked about the role of motor neurons in terms of how the muscles contract. And we basically stopped here. So where nerves are connected to the muscle fibers, to the cells, they're actually attached to those muscle fibers like that. This is the structure you saw before. Inside are myofibrils, and this is the sarcolemma. So they are actually attached, their ends are attached to the sarcolemma in those points which are called neuromuscular junctions. So where the attachment point is, their ends, those synaptic ends of those motor neurons, just like hands with these fingers or these pores of the fingers, that they are touching those cells like that. They're sitting on them and they are touching them at several different points. And when they're doing that, what they are doing is basically, which is shown in more detail there. But that whole junction where they are attached is called neuromuscular junction. So muscular is this and neuro is this. So this is where they are attached to. So within that, there are several terms that are described here. This is presynaptic terminal. This is the end of this motor neuron, which is called presynaptic terminal. And then they join together, which is here in this area. They are attached in this kind of indented place on sarcolemma of the myofibril. So this surface is myofibril that have mitochondria inside. There's a little bit gap left in between where the terminals are and where the cells are attached. So this is the sarcolemma of the cell. Again, this is the end of the motor neuron. And motor neurons carry this chemical signal with them. They carry neurotransmitters. Neurotransmitters are kind of moved here through this axon. They are moved here in this area, which is the presynaptic terminal. This is where they are kind of linked here. So this end is called postsynaptic membrane, which is the membrane of sarcolemma of the myofibril. Okay, so this is the whole unit. This is the whole neuromuscular junction, which is also called as synapse. So synapse is another name of that. and this is the connection between the nervous system or the neuron with these muscles. So all these terms are the same. Synaptic vesicles are those tiny vesicles which are contained inside the motor neuron, and synaptic cleft is that area in between, the space between the two connections, the end of the motor neuron and the beginning of the sarcolemma, and the postsynaptic membrane is on the side of the cell, which is the myofibril in this case, and presynaptic terminal is the end of the nerve cell. So this is an actual micrograph of how these axons branch and are connected to the myofibril. You can also see the striations there as well. So and within that area, where the connections are, this is where a lot of activity is taking place. That lot of activity that is taking place enables these myofibrils to contract. Neurotransmitters, which are carried by the motor neurons to the neuromuscular junctions, are kept inside. Neurotransmitters in case of muscle fibers, skeletal muscle fibers, are acetylcholine. So acetylcholine, or ACH, is the neurotransmitter which is used by the brain for contraction purpose. And this is the motor unit. Motor unit is a group of muscle fibers that are single motor, that are attached to a single motor neuron stimulus. So all the muscle fibers that are stimulated by the same motor neuron is called a single motor unit. So several bundles that are part of this motor unit in this case. Because a single motor neuron is not attached to only a single bundle but multiple bundles, and all of them are collectively called as a motor unit. Alright, so if it was not complicated before, you will see some more complications here now. So in this case here, it is showing how the actual potential actually plays a role, how the stimulus is received and how it is carried out inside. So in the previous slide we saw that these vesicles, which are stored here, these vesicles are stored here in the presynaptic terminal. So these vesicles don't empty by themselves. The signal to empty those vesicles here comes from the calcium. So where is calcium produced? Calcium is produced by the cell, by the myofibril, by the muscle cell. So muscle cell produces calcium, and calcium is always stored in sarcoplasmic reticulum. So sarcoplasmic reticulum releases calcium. Calcium is brought inside this presynaptic terminal. So the channel allows calcium to go inside, and when it goes inside, it actually stimulates to empty these vesicles. So those vesicles basically dock themselves against the walls, and they are emptied in the presynaptic cleft. The synaptic cleft is where those neurotransmitters that are contained here are emptied here. Neurotransmitter binds to these channels. These are sodium channels. So when it binds to the sodium channel that was closed before, so now this is inside the cell. So those channels allow sodium to go inside. When sodium goes inside, what happens? It becomes depolarized. When the sodium, in response to this signal, calcium to those vesicles, to the binding of acetylcholine, to these receptors which are present on the surface of this channel, these gated channels of sodium, sodium goes inside and causes this cell to be depolarized. Okay, so these are the four things that are shown that actually start the contraction. This is the first stage of the contraction, where the signal is received, and signal received here through those motor neurons is carried. Since there is no direct connection between the chemicals that are carried by the neurons and these cells. So those chemicals cannot go inside. Neurotransmitters don't go inside the cells. They actually they carry the signal in the form of some receptors that are present on the surface of the cell. So those receptors transmit their information to inside of the cell. Otherwise, neurotransmitters don't go inside. After the signal is transmitted inside through these receptors present on the surface of the cell, these neurotransmitters are taken back. They are either degraded or they are taken back inside the motor neurons. Alright, so this just talks about the same details, axial potential that travels down, causes the calcium channel to open. Then once the calcium channel opens, it goes inside, and actually it goes inside and causes those synaptic vesicles to release the neurotransmitter. For skeletal muscle, that neurotransmitter is acetylcholine into the synaptic cleft, and acetylcholine binds to the receptors present on the sodium channels, and sodium channels open. Sodium channel opening of sodium channel depolarizes, so it becomes positively charged. The cells, the muscle cells become positively charged inside, condition which is called as depolarization. So then depolarization, which is caused by the influx of the sodium, causes sarcolemma and T tubules to increase the permeability of sarcoplasmic reticulum, where sodium is released. So sodium binds to the troponin. So now another kind of series of events starts after that. Depolarization has taken place. The charge has become positive inside the cell. Then more calcium is released inside the cell, and calcium binds to the units of sarcomere now. So one event has already taken place. Depolarization has been completed. Now inside the cell, which is depolarized cell now, calcium is released. Calcium binds to the protein, troponin protein, and troponin protein interacts with tropomyosin protein, and then this helps to bind to the myosin. So this is kind of a chain of events that is taking place. Calcium to troponin to tropomyosin, and then to myosin at the end. Myosin then binds to the actin protein. So you can, if you want to actually draw or try to make a concept map, you can actually see what's happening there. Initially, calcium that was released was to cause the depolarization. The second event that is taking place with the calcium again is calcium in this case is binding to what protein? Troponin protein. And then troponin protein binds to what?
Actin.
No, one more protein.
Oh.
Tropomyosin. And tropomyosin binds to what? Myosin.
Myosin.
And myosin.
Binds to.
So that pretty much completes pretty much all the protein that we have seen so far. Actin, myosin, troponin, tropomyosin, all of them. So now actually the stage is pretty much set for that interaction that is taking place here. If energy is available, then the muscle will contract. So if energy ATP is available at that time, muscle will contract, because all of these series of events have already completed, in which several of these proteins take part in this process of interacting and activating other proteins with each other. So when ATP is available, then ATP will be released from the myosin heads, those heads that are touching the actin proteins, and the interaction of those myosin protein will actually bend in such a way that they will interact with the actin protein, and actin protein will contract with myosin protein in relation to myosin protein. So now that is the kind of action that is taking place. Myosin protein is pushing the actin protein, and all the sarcomeres within a muscle fiber will interact at the same time. They will contract at the same time in unison. So once this is done, once this one stroke is done, then that actual potential that was received from outside the cell through the motor neuron is completed now. The acetylcholine that was released will be taken up. It will be broken down with the help of this enzyme, which is called acetylcholine, just the name of the neurotransmitter, and it's acetylcholine esterase. So this is it's an enzyme that will break it down, because now the actual potential that was sent, the stimulus that was sent by the brain is removed from there in order for all the system to go back to where they were, to it got depolarized. So now once this acetylcholine is removed from the system, then it will go back to the same condition, which will be repolarization. It will get repolarized. It will change the charge again, negative inside the cell and positive on the outside of the cell again. Okay, so let's see those events again. Okay, so what's happening here is, maybe you can describe what's happening here. What is this thing?
That's the sodium, right?
Try it again. Calcium.
Calcium.
This is calcium.
Okay, I'm sorry, calcium.
So calcium. So when the signal is sent, this actual potential is sent by the brain, by these motor neurons. So that actual potential is in the form of these vesicles, which are stored here in the neuromuscular junctions on the presynaptic side. So calcium influx, the flowing of calcium inside this area will open these vesicles in this synaptic cleft, right? So this whole structure is neuromuscular junction or synapse. This whole thing is called synapse. So so these opening and release of acetylcholine, or this neurotransmitter which is contained in these vesicles here, what will happen is this acetylcholine will do what? These yellow ones,
The yellow ones will combine with the sodium.
they will bind to this channel, which is the receptors. This is the receptor for acetylcholine. So it will bind here, and it will open this channel. So this channel is for what? It is sodium. This time it's right. So first time it was not, but this time it is right. This is sodium channel. So sodium will go inside this muscle cell here. Muscle fiber will receive this sodium, and what will happen to the cell? It will get depolarized. It will become positively charged. So once it's positively charged, then in the next stage it will be, so it becomes positively charged, right?
Yep.
Okay, so this is stage
5.
So acetylcholine is released, calcium is inside. So acetylcholine stimulates sodium channels on the postsynaptic membrane to open, so it is inside. And what happens? So after depolarization is done, step number 5 starts. Step number 5 is sodium diffuses into the muscle fibers, initiating an actual potential that travels along the sarcolemma and T tubules. So that actual potential, the change in the charge inside the cell, will cause more calcium to be released. Calcium is stored inside cytoplasm, inside sarcoplasmic reticulum. So it is released from there, and it is carried on through those T tubules. T tubules will release calcium, and release of calcium will bind to several different proteins. It will bind to troponin, and from troponin to it will activate tropomyosin. Tropomyosin will activate myosin, and from myosin will interact with actin protein. So all of those things are taking place at the same time. It is, all those proteins are located here. So the calcium, the binding of calcium to troponin, which is released from here will cause all other interactions to take place. Okay, so once those interactions have been completed, now the next thing is the interaction between actin and myosin with the actin protein, and the conformation or the structure of myosin will change in such a way that it will actually bend. When it bends, it is removed from the actin, but it has already kind of pushed the actin towards itself. It has pushed towards the M line inside. So after this thing is done, after this one contraction is done where myosin has basically pulled actin protein, then the acetylcholine is removed from the system and it starts to go back. It becomes depolarized again. Once acetylcholine is gone, then it goes back to being the depolarized, to become repolarized. Let's see this animation. That actually explains all these events.
Action potential introduced at the neuromuscular junction is propagated along the sarcolemma of the skeletal muscle. At the T-tubule, the action potential is propagated on the T-tubule membrane into the interior of the muscle cell near the ends of the sarcoplasmic reticulum. The depolarization of the T-tubule membrane causes nearby voltage-gated calcium ion channels on the sarcoplasmic reticulum to open, resulting in an increase in the permeability of the sarcoplasmic reticulum to calcium ions. Calcium ions then diffuse from the sarcoplasmic reticulum into the sarcoplasm. Long filamentous tropomyosin proteins lie on two sides of the actin myofilament, covering up the sites where myosin can bind to the actin myofilament. Globular troponin proteins are attached to the tropomyosin. When calcium ions bind to troponin, this causes troponin to change conformation and move the tropomyosin. When the tropomyosin moves, this uncovers the myosin-binding sites on the actin, allowing myosin heads to bind and form cross bridges. The cross bridges play a role in muscle contraction.
See, the whole thing shifted, whole thing moved towards the M line, which is where these myosin filaments are actually oriented towards. So they can actually move only in one direction. Either they will move and they will come off, and then they will do the same thing again. When energy is available, they will bind to ATP again, and they will do the same action next time. And there are several ATP molecules consumed in that process. One movement of a single muscle will consume large amounts of ATP molecules. That's why the number of ATP molecules produced in this cell or other cells at a time are in more than 1 billion cells produced of ATP, more than 1 billion molecules of ATP produced, and